is, are there pieces that already suggest themselves to you as ones that would be that you're interested in having in an Excel file?
I'm pulling up the documentation now. To be honest, I can't remember off the top of my head what the different fields were.
Well, there's like the station, and then there's the date, and then everything, like the main part of the data is the time, and there's like different times with precipitation.
Yeah, how much they
Okay, so I would suggest that as a task, as a start for next time, I already have ideas what would be the relevant information, but I think it would be it's best for you to look at the documentation, look at what each line looks like, and try to figure out which fields you want to put into which cells, say, in an Excel spreadsheet. Does that make sense?
Yeah. We just need like a station number, date, time, and precipitation.
Right, and so you want to identify how are you going to get, because as I said, I've got code so far that splits each instance out. In MATLAB, it reads in the text, and then it'll split out each line, character by character, because that's how it's inputted into the data file. And if you can, so if you could, each of you say, email me which characters in each instance you would like to put in which cells.
Okay.
Because that's going to be, once we get it into an Excel or e-commerce separated value spreadsheet, then we'll be able to actually work with the data. This is kind of the hardest part, is at this first stage.
Okay.
All right.
Does that sound doable?
Yes, sir.
Okay. So with that in mind, let's go to the Adams State group.
I had a question. I wanted to know more specifically how the genes are related to whatever it is that we're trying to find, like age, like maybe race.
So in our, you mean in our data? Okay, so let's pull up that data and let me.
Who's on 479 here?
You, James, Katie, and Pablo.
We the boys and Katie.
So I'm going to bring up, instead of that data, I'm going to bring up the data for the NIH. So if you go to, we got the soft file eventually, didn't we? And it's on the computer. We downloaded it
Yeah. You did.
for an example.
Go to downloads.
You did.
And there it is. Okay, so if you take a look, was that maybe it wasn't the soft data.
That's one we got.
That's one of them.
Yeah. Because then you take.
We had to unzip it.
Yeah.
We unzipped it, but I'm trying to, so you're saying, what do all that relate to? So good question. So in essence, if you think about the discussion I had with Embry-Riddle, It's trying to understand what the data looks like, what pieces are going to be relevant there. And I mention that because you're gonna wanna really look at the Embry-Riddle course data carefully at what each field represents, because there is also another section, there's some sections that they have to be really careful about reading there. I'm not gonna give more away at this point, but with that in mind, let's take a look at the NIH data, and this came from, so for example, well actually I think I give it away in the project. Where'd it go? Description.
That was 479.
Yeah, but I just downloaded it again rather than go to
Yeah, she's in 479.
They're both the same, and where did I,
Right there.
right there. So first of all, so you're in the 479 group. So here is essentially, I give you what it's going to represent here, and there's two things. First of all, it's going to classify in one of these groups here according to these different probe settings here. And I think the zeros are the young. So you're looking at age differences as well as which section it corresponds to. For example, ocular motor versus spinal cords. So part of your task is going to be downloading, so if we go back up to the instructions here, a tab delimited matrix with row and column names, and you don't know what the probes represent. Microarray probe names, those are going to be your features. Those are your GSM,
GDS 2639.
whatever the numbers are. And so those are going to be your features, and they're going to be represented as rows. And the values, the sample codes, the cases and specimens for each one as columns. And so we want to identify young versus old in each of those four groupings. Does that answer your question?
Somewhat. I guess what I'm asking too, included in that is okay. So we're looking at the genes to specify whether or not from the data whether or not they have that specific gene when they're old or young.
Right.
So.
Well, I mean, they're going to have it, but it's going to be at different values, right?
Oh, so it's the values that we're looking.
It's the values that we're going to look at. The zeros and ones that you saw in that down below, that comes about. So if you recall, so I think I went through this. We have this gene expression data, and one of the tasks that we're going to start with, we're going to basically, if we, where did that go? I had the this data here, right? And so for each ID reference point, so for example, the first one in this row, and this is all one big long row, you're just not seeing it here.
Can you switch
Yeah.
the paper to landscapes? Do you think that help at all?
Sure. Would that be under,
I don't know if you can do it.
I'm not familiar with this version, so.
Well, I don't think you can do
You
that.
can't do that in
Word
Word.
pad.
Right, this is this WordPad? Yeah, this is WordPad. So, for example, at Sumo 2, this first value of Sumo 2, right, that I just highlighted,
Okay.
okay? That corresponds to the probe GSM 74342, and at that point it has a value of 1549.6. Okay, you with me so far, Katie? So I actually gave you a little more description here of what you're going to do with the Moses data. So so for that particular gene, Sumo 2, we're going to first average, if we see another Sumo 2, so there's an averaging value for each sample so that the features are unique. So if you have multiple values for that sample of that particular gene, you'll average those. Okay? Then the next thing you'll do is down here is you're going to take the base 2 logarithm of that value, the averaged value.
Okay.
And that's just a transformation into a narrower range. And it's standard in microwave data. And then, for use in Moses, not for use in the Weka group, what we will do, and I haven't had a chance to check Moses yet, but I'm pretty sure that it's still the same. I don't think continuous data is supported yet, but so what we'll do is we'll transform those values,
The log-based 2 values.
the log-base 2 values, by what's called medium normalizing, which means that each value will be replaced by 0 if it's under the median value over all the samples. So we've got different samples for that Sumo 2 gene. So for those, so we find the median value, and for a particular sample, if that value is below the median, we replace it with a 0. If it's above the median, we replace it with a 1. So that's how we're going to turn this continuous data into binary data.
Okay.
Does that make more sense now? And so now, just with those binary values, the question is, can we, in other words, below the median versus above the median, can we identify, classify samples as belonging to this particular?
What if it is the median?
If it's exactly equal to the median, then you could make pretty much an arbitrary decision which group it's going. Be consistent, but which group it'll go to. I mean, that's a question, right? How are you going to deal with cases like that? Because we only have if I'm correct with the current implementation of Moses, if it has to be binary, well, it's either a 0 or a 1. So it will be a little tricky at that point. It'll be a little bit biased, but it shouldn't, that's gonna happen so rarely that I wouldn't worry about it. So this comes to the point, just as I was mentioning for the Embry-Riddle group, the first point that you really want to identify is exactly what your question was, Katie. What does the data look like? What is the problem that we're trying to solve? And how does the data need to be transformed for use for particular algorithms? So in the case of the Embry-Riddle group, going back to pardon?
Oh, I'm just trying to move into my head what we're actually doing.
So in the Embry-Riddle group, and so I'm going to try to go back and forth here just to get everyone on target here. I don't know, did I download Colorado.txt?
I think so.
Or one of them, maybe it was Florida. I don't see it, so I'll do it here. Maybe I just showed it here, okay? So here's the Embry-Riddle data.
Holy cow.
Yeah, I remember that.
Right.
Yeah, but like the first 4 lines or 5 lines, I can't remember, they don't even need to pay attention.
Well, no, it's not the lines, it's the,
Columns.
so we have to be careful here because the state code, remember their task, so the state code's embedded in here. So when it comes to the testing protocol, they're gonna have to be a little bit careful that they don't put the state code as one of the fields of the testing protocol, right? Because that's gonna be their mission. So they're gonna just specify that as the class to be discovered, right? Within WCA. And that's easy enough to do. But part of this is, if you take a look, I wonder if I can make this larger here.
I think if you hold down Control and zoom, or use the mouse wheel up, it should zoom in.
Is that as big as it gets?
Possibly.
It's 125. It should be larger than that.
Or-
Zoom 150.
So it's just going really slow. Oh,
There we go. Okay, now it's kind of on its own. So if we take a look, so this is the Embry-Riddle data. At a particular line, a lot of this is not as straightforward, but the first 4, I can't remember what these first 4 aren't included. These first 4 characters are not included in the information in the documentation. Because if you go to the documentation, the first 3 fields are this 15M, which represents the kind of data, the 15-minute data. So every 15 minutes. I think the next 2 were, for example, the state code, which ultimately will be held out if you choose that as a class out of the testing set, but will be used in the training set, right? And now there's some other information here that I want the Embry-Riddle, I wish I could have a mouse pointer here. So if you go farther in here, this is what I was asking Embry-Riddle about, identify the particular characters that are going to be important for identifying precipitation records. And there is R2 right here versus right here. Okay, actually, it's these 2. Notice HT versus HI. And that represents tenths of inches versus hundredths of inches. So that's going to be important for them to handle the data, right? Because if it's measured in tenths, it's going to have to be modified versus hundredths. Pardon?
It's just I couldn't spot that myself.
Right.
I didn't see.
So that was what I was alluded to earlier when I mentioned there are some hidden fields that the Embry-Riddle group needs to be cautious about. Is how is everything being measured? There's a field, and it's in the documentation, and that's how I knew about it and looked for it. So.
Right.
So, in the Embry-Riddle's first task is identify what pieces that we need, and then, because the first step's going to be changing it into a comma-separated value spreadsheet. Because before you have that, you don't have anything. So, I don't know, is this working, going back and forth between Embry-Riddle and Adams State?
I've got a question for 379 and the class in Florida. Are we going to be transforming everything into a .cbs file or a .arc?
Well, WECA will use .csv. And if you have it in a .csv format, it's straightforward anyways. If you did want to translate it yourself, it's straightforward to change a .csv into an ARF file. And in fact, there is code within WECA that does exactly that. It takes a straight CSV file and will basically filter it in, uses a filter to read it into WECA. So if we have a CSV file, we're pretty much done.
Okay, well, I was just wondering, like, if you wanted us to go the extra step, we could set up the ARC file.
If you want to, it's not necessary if the filter works correctly. If it doesn't, then you should know how to transform the one format into the other format. But it should work just out of the box. I mean, that's what it's there for. If we wanted to go to WECA, I don't know if you want to see where that is.
Sure.
Okay. So WECA should be in this download is, oh wait, it's in a different location.
That's the installer.
That's the installer.
But you can just run the one off of the desktop,
Okay.
because the only thing that we needed that other one for was.
Was the package loading.
Yeah.
So. Now I don't know if I don't have a CSV file here to actually do it. That's the main problem. But if I go to this, the WECA Explorer open file, down here at the bottom of this open, you notice that it gives you options. The default is a .arf file, but you can also do, down here it says CSV data files. And it basically runs a script that changes those CSV files into the appropriate ARF files and then works with those. I wish I had a CSV file to work with. I don't know if I don't think any of the data files. Was it under x86 or was it?
Program files. Oh,
Doesn't matter. As all I'm looking for is the,
data folder. Oh, account. I bet that's all ours.
that's all our format. And I'm just looking to see if we had a CSV format. But let's just not get ahead of ourselves,
Okay.
right? Let's put it into the CSV format. If it doesn't work, we'll work with it then. It should just work out of the box.
What's R stand for?
Anybody want to answer James's question? I mentioned it, was it at the beginning, near the beginning of the semester? It's a file type.
Say we let him live with his confusion.
What's the A stand for?
Attribute, I forgot. He's gonna fix that.
Pretty sure I have it. Oh.
Attribute. Oh, would be a good quiz question, huh?
Relations.
What is it?
What is it before it defines it?
I don't remember.
Trying to remember Is it relation? Record. Attribute relation file format.
Oh, okay.
That's really, I got that right.
Attribute relation
I
file.
knew it was attribute something.
So, where were we? Getting back to on task. Does that answer your question?
Yes.
And I. Like examples, no? Where did the? So the other thing, and going back to, I'm gonna try to go back and forth between the Embry-Riddle data and the Adams State data. So now the other question is, that you wanna ask on Embry-Riddle, you've got these symbols here, and then eventually you get the times. And the other thing I really want Embry-Riddle to students to think about is how am I going to notice we've got the time 1945 and then a value. And you're going to want things, all your attributes to be lined up. So the question is, how are we going to organize that? And that's one of the things I want to hear back from Embry-Riddle students on Wednesday. So, let's see. Going to the, what happened to, thought I had the NIH data up here too. So going to the NIH data, well, no, it was the soft file. It was the,
Yeah, and you've got it open in your.
right. And WordPad is there.
That one.
So in terms of, I think that this part of it, it'll be harder on the second parts, but I think this part might be easier on the Adams State side. In terms of, but there's a lot of processing that still needs to be done, such as finding the averages and then taking the logs. And then for the Moses group, finding the median and determining how to adjust that. So, I don't know, are there questions at this point? I mean, this is all about you now, and less about me, but I'm gonna be here. So here's what I expect moving forward during the class, is every time you come with questions and difficulties that you have in getting the data to where you need it to get to. Yeah, Pablo?
Moses is installed on computers in Turin. Is that right?
Which computers, is it installed still on only the middle super
Yes,
computer?
I've got Balthazar updated to 14.4, but it's can cause some other issues, so I might try and just do a fresh install.
Okay. So far it's on one of the supercomputers.
Okay. And if you want to.
The other thing, Jeremiah,
Okay.
I thought about is it might be worth trying to play around and see if we can install it on, because it's a Dockerized, containerized virtual machine, right? And so it can be installed also on the Windows side
Yeah.
as a virtual machine, as a Linux virtual machine on the, even on the Windows side. So that might be worth a shot
Okay.
as well.
Yeah.
I can, if I ever have a chance, which I might have this afternoon, I might be, because I have a meeting at 5, so I'm gonna be around this afternoon. I'll take a look and, that I can maybe try on the, on Gia's machine, because that won't interfere with the Linux side, it'll be strictly on the Windows side, and then we can also use that. So now, as I said, here's the important part. It's your projects. I'm gonna be here to help, both Embry-Riddle and Adams State, but I can't help
You don't ask questions.
without you actually starting to dig in
Well,
and play around.
I think that I still have a hard time with what it is that we're looking for, I guess, because I rewrote what you said, basically, we are trying to identify genes that are more or less present at a certain age group,
Right,
old or young.
right.
And to me, that's kind of hard to identify, so I guess I'm trying to find a way.
Well, in essence, what you have, if you actually go into the geo database there, you're getting you're getting very minor variations in the values between, and it's kind of a spectrum of values. The values for each gene. And so from those values, what you're trying to do is, can you identify that this set of feature values comes from a young instance versus an older instance?
So if I went into,
And it's not just a single value that's going to determine that, it's more of a spectrum. Let's put it into maybe terms that you and I can understand more readily. Human aging, right? So what are some kinds of measurements that would differ a little bit, maybe not hugely, between a younger versus older human being?
Bone density?
Bone density, heart rate, maximum heart rate,
Skin.
skin.
Hair color?
Cholesterol.
Right. But do any of these guarantee, are these necessarily by themselves a guarantee that one comes from an older person versus a younger person? No. Does that put it in a little
well, the
better,
reason why I'm
or healthy
asking-
versus sick?
The reason why I'm asking is because I do know a little bit about genes, and in general, genes, you have genes, no matter what age you are. You have certain genes
Right.
inside of you, and it's usually dependent upon race or ethnic groups or stuff like that.
And these are coming from specific parts of the body.
And that's why I'm having a hard time understanding the old versus young, because no matter what age you are,
So let's go,
it's present. That's why I'm having a hard time.
right, now, good question.
Don't some genes degrade as they get caught?
So if I look, and I'm trying to see
I
where
don't
I
know.
saw this last time.
So that's why I'm
I
wondering.
don't know, I guess.
Um,
something that we'll look at in our research.
There was, I'm trying to, that went the GDS browser. What was it, GDS, let's look up GDS 1280. 'Cause that's one of the groups here. Search. And what all did we see there?
Skips this one. Pretty straight. Skips this one.
That's not what I was, I'm trying to remember where I saw this, because I started playing around here.
It says something about the underlying divergence effects of age on the central and peripheral nervous systems,
Right.
which is like, is that somehow a gene? Or is that just a nervous system?
Comparison of gene expression in the ocular motor nucleus. So that's where all these probes were put in the spinal cord.
Okay.
Animals 6, 18, and 30 months of age were examined, and so they're looking at the gene expression in each of those 3 age groups.
So basically, it's possible that it does degrade over time then?
Right, and I know that there was a spot here, I thought it might have been under data analysis tools.
Cluster heat maps.
Yeah, here we go. So I think this is what I was looking at. So here, if you take a look at what's going on here, here are, in the green on the left side is spinal cord, on the right side is the ocular motor nucleus. And in the purple, they each go 6, 18, and 30 months. Okay? And then you've got the samples, GSM 774 342 comes from a 6-month spinal cord. And there are slight variations. So let's take a look at, say, spinal cord, the first column here on the left versus, and so that's a 6-month-old value, and then compare that to the
Does that say 50 or
30-month values over here on the right. There are slight variations, and you're beginning to see the slight variations. More in terms of the spread of the data. I don't know if you see that. I mean, it's hard to see ourselves,
Yeah.
and that's why we're using data mining techniques to try to determine this. But you can see that there is a smaller range of values on the 6-month. So we've had 3 particular probes here on the 6-month. Compare those to the 3 on the 30-month.
Yeah.
And the median is pretty close to the same, but it's a slightly different distribution of values.
Okay. So my other question after seeing this, for each different gene set, the spinal cord, and then there was like a HIPAA or something.
Right,
So-
all of those.
Do they have different time frames for each set? Because
Yeah.
this here has the 6 months.
30-month.
Yeah.
So I don't remember the GDS numbers that we wanted to look at. It's on
In
the,
that Word document.
this Word document? Yeah. So let's look at another one. So we actually have 3 of these. GDS 2231. I pretty much looked at this 1280 a lot, and I'm also not a biologist, so I don't know.
Right, that's why I was really confused, because the only thing that I know about genes is just that.
So here, notice we have 6, 18, oh no, that's the same. I thought I got a new one.
Okay.
I need to
it's
search.
opened in-
So here, GDS 2231, aged marrow-derived stromal cells response to dexamethasone. So 3 or 15-month-old animals provide insight into molecular mechanisms regulating MSC differentiation in young and aged animals. So again, we're looking at this young versus aged. And the key thing here, again, let's go, I think it was the experiment design and value distribution curve here. Again, here we have 3 samples, 2 samples from 3-month untreated and 1 sample from the treated versus the 15-month untreated versus treated. And again, notice that there are variations. And they're not, in other words, the distributions are different. And you have to remember, these are log-base 2 values. So what that means is small variations are actually larger variations in the larger distribution, the unlogged values.
So these are already logged.
These are already have the log-base 2 here.
So the reason why we're averaging and logging is because of the different the different data sets having different times and
I-
whatnot? Or-
I mean, I really am not an expert in that field, why they use log-based 2
Okay.
for microarray data.
Okay.
Yeah, it's a convention. It probably works well that way. I wish I could really speak to that, but
Okay.
I have to let you know, I don't know everything.
What?
Okay, so,
Does that help answer the question, what we're, because it sounds like you've had more of a background
well,
in gene
I-
expression than I
I took a little bit
do.
of biology and some anatomy and stuff like that.
She's a stud.
No, I'm not. Anyway.
Not that.
Anyway.
So I was just really confused because I didn't know that it was possible for genes to degrade over time.
I've been waiting for someone to say.
So,
Yeah.
I mean, even when I taught the bioinformatics, we did a completely different
Hmm.
area, which the students voted on, and
Well, that kind of explains why they use like the
huge
stem cell
field.
research and the other
right,
stuff.
and they use the young cells,
Yeah. So,
and yeah, no, it's a
No, I have-
they're fascinating and huge problems right now in the biological sciences involving data mining. So, I don't know. So hopefully, hopefully that gave both groups, or both the Embry-Riddle group and the Adams State University group, a better feel for what it is you need to do. And really the first thing you need to do is like dig into the data. Try to figure it out. Because without starting that, you're not going to get anywhere.
yeah.
And then, you know, actually start the process. Questions in Florida?
I don't think we have any questions, but we've scheduled to meet up tomorrow to discuss the data and pick through it and try to figure out what fields we want to have set up in that document. And then once we can get a CSV file out of this data, then I think it'll be relatively easy for us to go into MATLAB and create kind of averages like that.
Okay, and as I say, I'm gonna be helping on that because your data's in a particularly not nice format.
Yes.
And I think I illustrated that by showing it to you, which is why I've started working on some MATLAB code to help you out.
You can meet, so we can know what it was. Actually, I think
Yeah. And thank you for that. That data, I looked through it quite a bit when I first got it, and it's like you said, it's really nasty.
Right. You're gonna both end up in some other difficulties as you go forward, but you're gonna find those out as you actually start working. So, okay.